pH and buffer are really important concepts in biochemistry. Biological systems maintain tight control on their pH and regulate the hydrogen ion concentration to really close detail. So we need to make sure we understand what pH is and how buffer systems work. The pH of a solution is the minus log of the proton concentration in a solution. You can also get to the proton concentration by taking 10 to the minus pH. So you should be able to interconvert between pH and the hydrogen ion concentration in a solution. Not sure why I can't page down. So let's think about this for a minute. Recalling that pH equals minus the log of H plus, which solution has a higher concentration of proton ions? A solution with a pH of 3.5 or 7.30? So take a minute to think about it. If it's 10 to the minus, the bigger number means that there's less of it. So the smaller number, 7.3, has more H plus ions. Another way you can think about this is pH less than 7 is acidic and pH greater than 7 is basic. Acidic solutions have more proton ions than hydronium ions, whereas basic solutions have more hydroxide ions or hydronium ions than they do protons. How about, can we do some math to figure out what is the difference in proton concentration between a solution that has a pH of 7.35 and a solution of 7.30? So to do this, you're going to need to convert the pHs to the proton concentrations, the H plus ion concentrations, and subtract and find the difference. Take a minute and give it a try. So if you would like to check your work, the proton ion concentration for a pH of 7.35 is 4.47 times 10 to the minus 8th. Notice that is smaller than, as we predicted, than the pH of 7.30, which we would say is more acidic, so it has more protons in it, which has 5.01 times 10 to the minus 8th molarity of proton ions in solution. The difference between those two, notice, is in the decimal spot, which is 0.9 or 10 to the minus 9th. So the answer is 5.4 times 10 to the minus 9th. Make sure you know how to use your calculator to do 10 to the minus x and how to convert from regular to scientific notation. That'll help you see the answers more clearly. The body controls pH very tightly, and there are two main buffers in the cells. The two buffers that we find in cells are phosphate, which is the main product from a lot of ATP-related reactions, and the other is bicarbonate, which is how we transport CO2 around the body. So let's review. What is a buffer? A buffer is a solution that contains a weak acid and its conjugate base, which is the acid with the proton removed, or a weak base and its conjugate acid, where the base has gained the proton, and that's called the conjugate acid. And this combination resists a change in pH upon the addition of strong acid or base. So it allows you to control the pH of the solution. This week in lab, we're going to be making buffers that we're going to use for the rest of the semester. So it's really important that we know how to do these calculations. So as a reminder, strong acids and bases completely dissociate. So if we put hydrochloric acid in water, it completely dissociates to form H plus ions and chloride ions. Weak acids and bases only partially dissociate, and in fact, there tends to be way more of the weak acid form than the ionic form present. So notice this is an example of equilibrium. It's drawn with the arrows pointing either direction. So let's review for a minute what an equilibrium is. So what is an equilibrium? When we're at equilibrium, does it mean that the reaction stops? Does it mean that the rate of the forward reaction equals the rate of the reverse reaction? Does it mean that the concentrations of the reactant and product are constant? Does it mean that the concentrations are equal? Should have made that D instead. Does it mean all of the above? Does it mean A and C? Or does it mean B and C? And if you want to review, PhET has a really interesting simulation that looks at reactions and rates to let you play with equilibrium values. take a minute and think about it. Which ones are true? What are true are B and C. It doesn't mean the reaction stops. It just means that the rate of the forward and reverse reactions are equal, which makes it appear like the reaction has stopped because the concentrations of the reactant and product remain constant. They basically tell us about the extent of the reaction. So we measure these with equilibrium constants, where we look at the concentration of the products over the reactants. And for an acid-base reaction, this is given a value called the Ka value. So notice we've got the concentration of A minus, which is the deprotonated acid, times the concentration of the protons in solution, divided by the concentration of the reactant, the protonated acid. So how can we rearrange this to be able to talk about pH of a buffer? So we can take that Ka expression and we can do 10 to the minus on both sides. So that is, we can separate it into the two parts. So the one term being the A minus over HA term and the H plus term. We take minus the log of each of those terms, which makes each of those a P term. So minus log of something is P of that. So it becomes pKa equals minus log of A minus over HA plus pH. And just because it looks nicer, we tend to rearrange this and solve for it in terms of pH. So we basically move the log of minus A over HA to the same side as the pKa value, because it makes it easy to calculate pH, and it also puts everything in terms of in positive terms. So notice this is really just a rearrangement of the equilibrium expression, but it makes it really easy to calculate pH based on the pKa and the ratio of the basic and acidic forms of the acid. So in general, buffers work best when there is a fairly equal ratio between A minus and HA. So notice if you look at the pH expression in the corner, when the pH is equal to the pKa, then the log of A minus over HA is going to equal 0. And the log of something equals 0 when the thing that you're taking 1 or 0 equals the log of 1. So when the concentration of A minus and HA are the same. So pH, in general, buffers work best when the pH is close to the pKa. And close, this is kind of like dancing or dynamites, where how close is close? Well, it depends on your system. In general, you want the pH to be within 1 pH unit of the pKa value. So if we consider phosphate, one of the buffers that we said was the main cellular buffer, we've got three choices. We've got the triprotic phosphoric acid, we've got the diprotic phosphoric acid, and the monoprotic phosphoric acid. And they each have different pKa values. So if we're thinking about which species are going to be found at pH 7.4, the one that's going to be most prevalent are the ones with the pKa closest to this. And if you've taken Chem 331 or are in there right now, you might actually calculate what the ratio of all these species are in solution as part of your analytical efforts. But in general, we're going to go with the rule of thumb of the one that has the pKa closest to the desired pH is the one to use, and you want it to be within 1 pH unit. So often when we're writing out phosphate in biochemical reactions, we'll just write Pi, because what's the protonation state? If we're at pH 7.4, about half of it is H2PO4 minus, and about half of it is HPO4 minus 2. So that's confusing. So sometimes we'll just write Pi as an acronym, meaning that it's a mixture of those two species. The other thing to keep in mind is Le Chatelier's principle, which talks about when we've got an equilibrium, if we change the circumstances, including concentration, temperature, volume for a gas, or partial pressure of a gas, then the equilibrium will shift to counteract that change and will establish a new equilibrium. And that's found throughout biochem, that we're going to be shifting concentrations to affect systems, and very small changes can lead to big changes in the overall system. So after going through this material, you should be able to define what is a buffer, an equilibrium, and Le Chatelier's principle. You should be able to convert between pH and proton concentration. You should be able to use Henderson-Hasselbalch equation to calculate the ratio of the species at a given pH and find the pH if given the ratio of the species. You should be able to define equilibrium reactions and use them to predict reaction directionality given conditions. And you should be able to explain how a change in a system is compensated for by Le Chatelier's principle. And in class, we're going to be looking at how bicarbonate is able to blood it for blood pH. We just looked at how phosphate works. If you have further questions about these, please go back and review your Chem 111 notes and or come and see me or the QSC tutors.